At the Division of Healthcare Quality Promotion, one of our primary areas of focus is preventing healthcare-associated infections, which include antibiotic-resistant infections.
Antibiotic resistance is one of the most, if not the most, important public health threat facing our generation. Antibiotics have done wonders for health and modern medicine. Any of these interventions we do rely on antibiotics that we know we'll have something to turn to treat a serious infection that so often occurs. Antibiotic resistance has really gotten to a crisis stage. Over 2 million infections in the United States each year and 23,000 deaths. In addition, there's another 29,000 deaths that are occurring in people who've recently had Clostridium difficile infection.
There should be a sense of urgency that we need to take action now. And microbiome indices we envision as a standardized way to assess the health of someone's microbiome. There's several applications for microbiome indices in the future, including patient health, antibiotic stewardship, drug development, and also assessment of treatment or intervention. In addition to preventing an individual from getting sick, we envision that microbiome indices could also be used to assess whether an intervention is working.
These indices will certainly also help, we hope, industry as they develop new antibiotics or develop a preventative. And that's where we do work with industry, where we have an alignment of interest between public health and the private sector. We're going to need new innovative approaches and really push back and make strides against antibiotic resistance.
The microbiome represents a huge amount of untapped data, and if we can actually leverage that data to better protect patients, we'll make a huge impact in our ability to prevent antibiotic-resistant infections before they even happen. I think the return on health that we can really benefit from here is that rather than treating someone after they're sick, if we can prevent them from getting sick in the first place, we can save lives, prevent people from getting additional medications. In the end, we can protect the public health and patients' health as well.
Synthetic Biologics, we focus on tangential products that come out of the microbiome, specifically the GI microbiome.
Our approach is aligned with the CDC's approach and with its goals, because our goal is to prevent C. difficile infection from emerging at the point of use of antibiotics is directly aligned with reducing the burden of C. diff and antimicrobial resistance, which is the primary goals of the CDC in those fields right now.
Ribaxamase and Synten are our two lead products. They're both going into phase 3. Ribaxamase focuses on fixing or preventing the occurrence of C. difficile infection, as well as the occurrence of antimicrobial resistance. Our second product's Synten, and that's for irritable bowel syndrome, constipation.
Both of these compounds have a clear regulatory path through the clinic and a very, very large and robust patent estate around each of them.
Ribaxamase, it's a new approach to the C. difficile infection problem. Rather than treating the C. difficile, we're preventing it, and we're doing that by inactivating the antibiotic that gets into the gut and protects the microbiome. So if the microbiome is intact, we don't have the C. difficile infection.
One of the great challenges of manufacturing ribaxamase is we cannot interrupt the action of the antibiotic systemically in the body. What we want to do is set up a barrier on the large intestine so that as the body processes that IV antibiotic and slowly secretes portions of it into the digestive tract, there aren't adverse effects of that antibiotic interacting with the gut microbiome.
We choose strategies that have simple yet elegant mechanisms, and we demonstrate efficacy with our simple mechanism in multiple animal models. Then we show that we can achieve efficacy in humans. So when all that falls into place, and we can be confident that we will successfully traverse all the way through clinical trials.
The underlying premise of this is that your microbiome is very specific for you and your general well-being. I think of this in a similar mechanism as a wildfire burning through a prairie or a forest. Once the destruction occurs, non-native species or weeds can fall in place and overgrow the native species. Our program, SYN004, is a fire prevention mechanism. That is to say, if the antibiotic doesn't get to the gut, the fire's never started and the destruction doesn't occur.
So as we're discovering today, the microbiome and disruption of that microbiome, the destruction of the diversity by using antibiotics has a profound impact on human health downstream. So from a public policy perspective, obviously we want to reduce those number of incidences as much as we possibly can. Ribaxamase is the ideal tool to do that. You're going to pop ribaxamase capsules the same time you take an antibiotic, and lo and behold, you're going to retain that diversity, that rainforest internally in your GI tract, which keeps you healthy.
I get up to work every day with a sense of urgency and pride in knowing that the work that we're doing here could potentially help make the lives better for millions of patients who suffer from C. diff and from IBS-C.
The team here is pretty amazing. We're doing great big things around the gut microbiome. We are passionate, we are protectors, and we are pioneers.